Hi class, in this recording we're going to focus on the anatomy of the ear, the inner, middle, and outer ears. So as we look at our ear, the ear is a structure that's designed to take a sound wave and transmit it as an action potential. So we need to take the middle, outer and middle ears, which are shown, here's the outer, and then here's our middle ear in this area over here. This tube is considered part of the outer ear. Let me redo that here. Here's our outer ear in green. And then here's our middle ear in blue. These first two portions of the ear are primarily concerned with taking that compression wave, that sound wave, and converting it to a signal, or converting it in such a way that can be processed by the inner ear. And this inner ear I'm outlining in red for us right here. And then within the inner ear, we take those vibrations and turn them into action potentials. So as we look at the outer ear, there are some structures we should talk about. For instance, we have the whole ear itself. And as we look at this entire ear, it's referred to as an auricle. The auricle of, in ancient times or in several hundred or several thousand years ago, when we think of oracles, an oracle is somebody who would be speaking the words of God to you. So you would really want to listen to somebody who was an oracle. And I'm thinking of oracle as O-R as opposed to A-U-R. Another term that's used to refer to the outer ear is the pinna. And I've seen these both used equally. So I don't really see one used preferentially over the other one. And as we look at this auricle, it's going to be supported by elastic cartilage. And then this auricle is going to have a hole right in the center. Let's shift colors to bright green so it's easier to see. So that bright green hole right in the center, referred to as the external acoustic meatus. As we look at the external acoustic meatus, also referred to as the auditory canal, it's going to take compression waves, or sound waves, and send them down into the middle ear. We have guard hairs in our ears, and as we age, these guard hairs unfortunately get larger and larger. I'm starting to get to an age where I need to keep them in check so they don't stick out of my ears too far. The guard hairs are going to focus on protecting the outer end of the canal, in particular to keep large items like bugs or insects from crawling into our external auditory or external acoustic meatus. And we can see some of those guard hairs as some white fuzz, located there within the external acoustic meatus. We also have cerumen, which is known as earwax. And as we look at this cerumen or crumen, as it's sometimes pronounced as, it's going to be the secretions of modified cruminous or modified sebaceous glands, known as cruminous glands, plus regular sebum from regular sebaceous glands, and a good healthy mixture of dead skin cells to give you that orange flaky substance that comes out of your ears. Now, once we get to the middle ear, our middle ear is going to begin at the tympanic membrane. This is the barrier that separates the middle ear from the outer ear. This tympanic membrane is also referred to as the eardrum. And the tympanic, or the middle ear, is going to be housed entirely within the temporal bone, along with the inner ear as well. And as we look at the tympanic membrane itself, it's fairly large. It's about 1 full centimeter in diameter and can oscillate back and forth. It can vibrate freely in response to sound waves hitting it. Our tympanic cavity. So this, let me shift colors here. This big, I was trying to get yellow. This big space right here, this tympanic cavity, is going to be continuous with some mastoidal air cells. And in terms of the total distance, it's about 1 to 3 millimeters wide between the inner and outer ears. It's a very, very small amount of space. Notably, it contains some auditory ossicles. When we think of an ossicle, think of the root word ossi for bone, and auditory referring to our sense of hearing. So the hearing bones are the auditory ossicles. As we look at these auditory ossicles, they have some old school names, the hammer, anvil, and stirrup. Um, and depending on how crusty you are, maybe that's what you had learned back when you were in middle school or high school. The names that have been phased in, though, are the stapes, incus, and malleus, from lateral to medial, or from external to internal. So as we look at this first bone that attaches directly to the tympanic membrane is going to be this, excuse me, is going to be the malleus. And then as we look at the next bone, the incus in green, that incus is located right here. And then finally, the stapes is located right here. So I misspoke just a couple seconds ago when I was referring to orientation. What I think is MIS. You can't miss the answer if you know that the malleus, incus, and stapes goes from outside to inside. MIS. Now, as we're looking at the eustachian tube of the ear, the eustachian tube is kind of an old school term. It's being phased out right now in favor of the auditory tube. It's going to connect the middle ear to the nasopharynx, or the back of our throat, and it allows for air pressure to be equalized on both sides of the tympanic membrane. It's important to equalize air pressure because we want the tympanic membrane to move freely. If we have a large pressure gradient across the tympanic membrane, that restricts the movement of the tympanic membrane and makes it harder for it to oscillate and for us to hear things. Normally, the eustachian tube, and I'm a bit older, so I'm going to say eustachian tube because that's how I was taught. The eustachian tube is going to be closed, but yawning or chewing or swallowing helps it to open up. This is one of the reasons why, if you're taking a young child or with you on an airplane ride, you oftentimes give them chewing gum during landing and takeoff to help them open up their eustachian tube and equalize the air pressure. If you have an infant that's breastfeeding, a lot of times the mother will breastfeed the child during landing or takeoff to help open up the eustachian tube and minimize the pain associated with those pressure gradients. And I already talked about the bones. We have the malleus, incus, and stapes from exterior to interior, or we could say lateral to medial. And directly underneath the stapes, it's worth emphasizing we have an oval window. This oval window is a membrane that marks the beginning of the inner ear. We also have two muscles, the stapedius and the tensor tympani. And these muscles are going to attach to, well, the stapes and the malleus and allow for them to be regulated. So they help to upregulate or downregulate the amount of sound that we're hearing or perceiving as our eardrum or tympanic membrane oscillates. If we look at the ear, the middle ear between an infant versus an adult, a pediatric eustachian tube is going to be relatively horizontal, whereas an adult's eustachian tube is going to be relatively steep and will have better drainage. And because the pediatric, or the small child's, eustachian tube is so shallow, a lot of times those young children are going to have otis media, or a middle ear infection set in. Media referring to the middle ear, otis referring to, when we think of ITS, that's inflammation. Inflammation of what? The ossicles of the middle ear. And then those young children oftentimes are going to have a lot of pain associated with that. And we really need to emphasize pain and impaired hearing. Occasionally, these ear infections can spread, leading to meningitis or fusion of the ear ossicles. For instance, my father had so many ear infections when he was a kid, he had partial fusion of his ossicles, and he lost 1/3 of his hearing within the first 5 years of his life because he had so many ear infections. And I started life with the same process. I had so many ear infections when I was young that by the time I was 4, I remember having tubes put in my ear. I remember this very vividly. And one of the cool things about having these tubes put into my tympanic membrane is that when I would plug my nose and hold my mouth, I could blow really hard, and push air through my eustachian tube to my middle ear, and then through the tube in my middle ear, and have bubbles come out of my external ears while I was underwater in a swimming pool. And I remember being a 6 or 7 year old boy thinking this was the coolest thing in the world. I would laugh so much when I would do that. As we look at the inner ear is going to have a bony labyrinth and a cochlea. Those are the two main parts of the inner ear. And as we're looking at the inner ear, the bony labyrinth is just a series of passageways within the temporal bone. And it's also going to be lined with the membranous labyrinth. So the flesh that lines the bony tubes is referred to as the membranous labyrinth. And there are going to be some liquids there. We have the endolymph and perilymph. As we look at the endolymph, in terms of its chemical composition, it's pretty similar to the intracellular fluid. And as we look at perilymph, it's pretty similar chemically to the CSF, cerebral spinal fluid. One of the things that is kind of cool is that if there's a sodium imbalance in these liquids, that can alter the pressure in the inner ear and the sense of orientation. There's a disease referred to as Meniere's disease that's associated with this sodium imbalance. I actually worked next door, across the hallway from a gentleman who developed Meniere's disease in his mid-20s. And it was not a fun experience for him, to say the least, because he was constantly nauseated and thrown up. He would be seasick while he was sitting still on his desk. We have a labyrinth. The labyrinth itself is going to consist of a vestibule with some three semicircular ducts. Think of X, Y, and Z axes when you think of the three semicircular ducts. And let's shift to red here. We also have a cochlea. This cochlea is a spiraling structure. It looks like a seashell, and it's going to have 2.5 coils that are going to wind around an axis of spongy bone and up around an axis of spongy bone. There we go. That axis of spongy bone is referred to as the motolus. And as it's threading around that, it's going to support some fleshy tubes of the cochlea that are going to take different frequencies of sound and turn them into action potentials that we can process. As we look at the cochlea, it has 3 fluid-filled chambers, and they're going to be separated by membranes. We have the scala vestibuli, which is the superior chamber, and it's going to be filled with perilymph. We have the scala tympani, which, let me shift to yellow here. Scala tympani is going to be the inferior chamber and also filled with perilymph. And then we have the scala media. Media, think middle for media. This is going to be the middle membrane, and it's going to be filled with the endolymph. And the names of these structures are a big clue as to what they attach to. So as we look at the scala tympani, it's going to attach to the tympanic membrane. As we look at the scala vestibuli, it's going to begin at the oval window and spiral to the apex, which means that it's going to start here at the oval window and spiral all the way to the apex, which means that it's going to go through the vestibule of the middle ear, which is this region right here of the inner ear, the vestibule of the inner ear. Hence this name, scala vestibuli. Scala media is a very straightforward name. It's the middle of the 3 membranes. Now, as we're looking at the spiral organ in our ears, our spiral organ is going to have a bunch of hair cells and supporting cells. Those hair cells can be long and stiff. Those ones, the stiff ones, are referred to as the stereocilia, and they are going to be on the apical surface exposed to the cavity itself. Attached to, what do we have going on here? So there's the stereocilia right there. Attached to those stereocilia, we have the pectoral membrane, which is a fold that goes and separates the two of the cavities from each other. And the spiral organ will have 4 rows of hairs spiraling along. So we have, to sound off here, 1, 2, 3, 4. 3 rows that are super close together. And then the 4th row which stands alone. The 4th row is referred to as an inner hair cell, and it'll have approximately 3,500 cells and be used for our sense of hearing. As we look at these 3 rows, these outer hair cells, there are approximately 20,000 of them, and they adjust the cochlea. They adjust the cochlea in response to different frequencies and increase the precision of our ear to take sound waves and convert them into action potentials. In terms of the physiology of hearing, let's think of the function of hearing. This is our simplified diagram of the ear. We have the outer ear, which is going to take sound waves and funnel them towards the tympanic membrane, and then that tympanic membrane oscillates back and forth. And it'll take, as we look at the bones of the middle ear, the malleus, incus, and stapes, those bones take the movement of the tympanic membrane, which is relatively large, about 1.5 cubic centimeters, and concentrates all that force into a tiny, tiny little space, the oval window. And then all of that force is going to take the fluid of the inner ear and cause the fluid to oscillate back and forth. It will push in at the oval window, and then we'll push out at the round window, which is kind of a pressure relief system for our inner ear. And then as that fluid oscillates, there's the basilar membrane of the inner ear that's used to move the tectoral membranes and generate action potentials from those sound waves. So as we think of the ossicles, they concentrate energy. And the ossicles are quite strong in terms of the amount of force they can generate because they need to move a thick gelatinous liquid, the perilymph, through the inner ear. And the ossicles themselves serve a function in that when there's very loud noises, those muscles that are connected to the ossicles can adjust tension and reduce the amount of noise that are vibrations that are transmitted to the inner ear and minimize damage from those loud noises. That's all we have for this recording. If you have any questions or comments, class, please feel free to post them on the class discussion board or to shoot me an email. And as always, happy studies.